Okay, so welcome you all today. Thank you very much for joining us. I will start with some messages for the participants. So for people that are watching virtually, so virtual participants can type their questions in the Q&A box below the live session screen. So you can start typing the questions as the session starts. So keeping up on that. And then during the questions, when you are selected to ask your questions, and this is for people here at the audience, please wait for the microphone to arrive and please hold it close when talking. So today is my great honor to introduce here our keynote speaker, Professor Jeremiah Faith. So Dr. Faith is an associate professor in the Department of Genetic and Genomic Sciences, Medicine and Clinical Immunology, and also a faculty member at the Ichan Genomics Institute in the Ichan School of Medicine at the Mount Sinai in New York. Professor Faith is a microbiologist and a system biologist working in the intersection of microbial ecology, genomics, and host microbe interactions. He was trained in quantitative and experimental methods to study the complex microbial communities and has been a pioneer in applying ecological principles and strain-level genomics to the human gut microbiome. Over the past decade, his work has fundamentally reshaped our understanding of microbiome stability and long-term colonization, and his group has demonstrated that specific bacterial strain can persist for a long time in the human gut, emphasizing the importance of strain-level resolution in understanding the microbiome and in health and disease. By combining longitudinal human studies and controlled experimental systems and quantitative modeling, Professor Faith's laboratory is moving the fields towards a more mechanistic-based microbiome therapeutic. And for today, he will be talking about the design, manufacture, and engraftment of live microbial therapeutics for infectious inflammatory diseases. And while I was preparing this introduction, I have to say that I came across with the information that besides being a great scientist, he's also a fine musician. So thank you very much for accepting our invitation, and please welcome.
All right, good afternoon, everyone. To all the American folks here, good morning if this is morning. Beautiful weather. I love the UK when the weather is good, so I'm happy to spend 2 days inside with you all. For the people that don't want to be inside and like to go running, there's always somebody at these conferences brings a pair of shoes. So I'm going to go for a run tomorrow morning. If you want to go for a 10K, 15K, let me know. I tried yesterday and it's super nice. You also, on my run, I found this as one of the biggest McDonald's I've ever seen. Bigger than even in Texas, so it's a good place for going for a run. Here's my conflicts of interest. Okay, so what I would like to do is to bring in some Winston Churchill, because I'm in the UK, it's a little bit cheesy, but why not bring it? Is microbiome research at the beginning of the end or the end of the beginning? And that's the thing I would like to talk about today. For those of you who have been here since the beginning of the microbiome, I'd like to say the microbiome is probably about 20 years old. I'd like to say I got into it maybe 17 years ago, so I don't feel too old. But really before that, it was anybody watch Star Wars? Star Wars has like the rebels, and sometimes in the movies the rebels are a lot of people, and sometimes there's little bits of people. And before 2005, the rebels were a small group of people that thought the microbiome might be important, and then it exploded with 454 sequencing. It became the most popular thing everybody's talking about on the radio. It's, you know, your friends are being like, oh, I know about the microbiome too. However, we're in the period where if we don't play our cards right, we could become back to the small rebel alliance. And what I would like to propose to you all is to start working more in humans, to do more interventions, because the more we make people not just talk about don't wash your hands, but you know, I'm taking some pill and that's making me not have some disease, the more that we will have a golden era. So if this is the beginning of the end, what will we find? We'll find that our first and most important accomplishment is the approval of 2 drugs for the treatment of C. difficile infection, namely Rebiota and Vowst. I think it's very likely that we'll also have a second accomplishment, which is a defined consortium, which is a little more sophisticated. We could say we have a proper drug. The third accomplishment in this scenario is we have lots of proof of principle that we can cure mice. Many of you have probably been involved in these types of things. We have a lot of human FMT studies that are too small to be definitive, but big enough to be interesting and intriguing. And we have beautiful discoveries in ecology. I think the last 20 years we have learned a huge amount about what's the structure of the microbiome, how does it change over time, how much can you manipulate it. In the previous eras of the microbiome, which were many decades ago, I think it was much more primitive than where we are now. The question is, can we use this as a foundation to grow and to develop new therapeutics? Or was this just a very expensive, fancy ecological discovery, right? The end of the beginning scenario, which is where I hope we are, is we have the same couple of accomplishments, but in academia we start to move away from what was very heavily collect a bunch of stool samples and see what's in them to and mechanistic mouse work to hypothesis-driven human trials. And some of the people in this room are working on this type of thing already. This would bring de-risk products to industry. So rather than having industry be the people that pioneer and figure out if stuff works, our job is to figure out if stuff works and then to give it to these experts at bringing things to the market. And I think we'll find much better reception if we show phase 1B really strongly positive trials than if we say, I got some microbes that make my mouse better. Can you see if it works in a human? This will lead to happy patients, glorious riches, and the golden age. The next meeting will be caviar and truffles will be all that we have at their lunch. I think we have lots of promising starts. There's a lot of indications where I would say, if I had 2 lives to live, I could go on any one of these and have a good time trying to see if this matters. And I would just encourage you to think about your basic science, but also can you have an impact in your current round of whatever you are, postdoc, PhD student, faculty. So our group studies the structure and the function of the microbiome with the goal of developing therapeutics. We don't do all 3 of these things, but I'm only going to talk about this one little scenario today because I'm trying to pitch people that by the end of this talk, you feel like you want to go home and build a manufacturing facility to give microbes to your friends and family. So this started for us working on C. difficile infection by following the work of Ari Greenspan, who was a young doctor at the time that decided to start doing fecal transplants back when it was a little bit crazy to be one of the people doing this. And what we found was that the microbes before the transplant from the donor wouldn't be in there, but after about a week, you would find that most of the donor microbes were there, and even with only a single dose, 5 years later, you find the same microbes, that we have this very high engraftment, and the engraftment lasts for up to 5 years. The other thing that he found was that the more the microbes get in, the less likely the person is to relapse. So from this, you can make a pretty simple sort of algorithm of like, how do you pick the best drug for C. difficile infection? Well, if more microbes getting in is an improvement, try to find the microbes that get in well. This has now been repeated in many studies, including, this is pretty small, our data on the left here, but the big study on the right is from the Rebiota trial. And now with hundreds of people, they find the same thing, that more microbes in predicts non-relapse. The other thing that we did about a decade ago was set up a culturing facility. And I saw there's a bunch of talks today about high throughput culturing and trying to do mechanistic work with culture, so I'm super excited about those. We set up this large anaerobic coy chamber, which you can see here is about 20 foot long, and inside of it we have a liquid handling robot that allows us to quickly isolate bacteria from different people. We use a MALDI-TOF to identify them, and we put them into 96 well plates when we're done. We make about 1 of these a week. We've made 200 or so libraries, and most importantly, I think we've tried to scour the world for the fecal transplant donors that have been used a lot. People that are good at giving stool donations and often pass those tests tend to be giving their stools to lots of people for lots of different diseases. So we've isolated as many of those as we can to use as raw materials. And the important thing about those microbes is by looking at them in fecal transplant trials, we can end up with tables like this, where we have each microbe, how often it engrafts in the recipient. So we can pick microbes that are not going to just run straight through the person and be flushed down the toilet, but that will go into the person and be able to grow and perpetuate. So our first design of our first drug was to take the microbes that engraft well and assume that engraftment is beneficial for C. difficile infection. And we used that to come up with a very simple algorithm to result in what our first drug was, which is called MTC01, or Microbiome Translational Center 01, the first of our 99 drugs. So here's the facility that we built for manufacturing. So this is our GMP facility. A classic GMP facility is like a clean room where you have to put on a costume and walk around in there and step on this sticky tape. We didn't go with one of those. What we did instead was we built a bag that's just an anaerobic chamber, and we put a sterilization port in the front of it that allows us to clean the chamber rather than cleaning a room. Made for much quicker turnovers, and actually is much cheaper than building a whole room, of course. We would grow all these microbes inside of this chamber, and this is work led by Lucas Bethlehem. So our first study is just to try to compare a stool sample with a defined consortium of microbes that came from that stool. So we had one human being, we isolated their best engrafting microbes, and we wanted to do a head-to-head between the stool sample of that donor and the best engrafting subset from that same donor, giving half of the patients one, half of the patients the other. This is work led by Ari, Sari, and Few. We wanted to just answer some very basic questions. Obviously, C. difficile has already been cured. We already have drugs out there, but for this type of trial where we can do a head-to-head, what can we learn? We can learn the safety and efficacy of FMT versus the defined consortium. We can see, do in vitro manufactured microbes work as well as microbes made in the colon? You would assume that microbes growing in the colon are the happiest because that's where they're growing up, but when we put them on a plate, does that make them like sick? And when you put them in the human, they don't do quite as well. Does this pre-selection of good engrafters actually pan out in a real drug? And our pilot trials of in-house manufactured microbes practical? And the answer to the last one is yes, because we're finished with the study. So this is what actually played out in the trial. We ended up with 2 doses. One dose was 10 to the 10th, one was 10 to the 11th. So people would go into one of these 4 groups, either stool low dose, stool high dose, LBP low dose, or LBP high dose. And the red people are the people that relapse. So you can see at most 1 per group relapses. And the numbers are in line with what we know from prior studies. Although our trial is obviously too small to have a concrete number here, we're somewhere in the ballpark of what's been shown with Valstri, Biota, and CP101, which are the 3 largest randomized placebo-controlled trials of fecal transplants. So how well do the microbes get in? So here we see the donor, the FMT, and the MTCO1, and the different doses. And 15 is the maximum number of strains because that's all the number of microbes that are in the drug. And you can see that the best one is MTCO1 high dose, but in general, they're all about the same. We get very good engraftment as we were hoping for. The other thing that you can see on the right side is that once the microbes get in, they stick around. Just like with the fecal transplant where they stuck around for 5 years, here we sampled out to 6 months, and whatever got in on the first week was there 6 months later. So that's all looking quite promising. If you look at the individual strains, you can see most of them get in. Each color here is a different one of the groups. The purple one you can see is always a little bit lower. The low dose of the defined consortium for some reason doesn't get in very well. And the other thing that you might notice is that there's 4 microbes where the FMT looks like it either never gets in or it rarely gets in. And what we think happened is those microbes were lost from the donor in between the period where we made the drug and then we had the donor come back. And it was about 5 years or so. And it looks like they lost those microbes. They ended up with a new strain of the exact same species that engrafts perfectly fine, but those 4 are lost. So I think this is a pitch for why defined consortium are important, because you could imagine your best drug or best microbe was in there, the person loses it, and then your donor comes from having like golden stool to having not very useful stool. But when it's in the freezer, you can just grow it back up. Okay, and then do microbes that are picked to be engrafting well engraft well when we make them as a proper drug? So on the left are all of the strains from the donor that were not in our 15-member consortium, and in the middle and on the right are the 15-member consortium, either directed by FMT or by MTCO1. And as we would expect, the microbes that we pick to be good engrafters engraft much better than the ones that are from the donor but were not selected. So each dot here is a different strain. You can see most of the strains that we didn't pick don't get in very well. So picking the good engrafters, I think, is a standard rule. If you think the microbes you want need to colonize, then why not start with microbes that we know colonize well? Even if you wanted to do genetic modifications and engineering of these microbes as a chassis, I think picking these types of microbes is really the way to go. Okay, and then one little piece of basic science that we didn't really pre-plan but kind of came out of the study, and after you see it, it's pretty obvious, but weren't thinking about this when we designed the study. And that's just looking at the overall microbes and not just at the 15 strains. And this is now looking at alpha diversity. And the dotted line here is the alpha diversity of the donor. And you see on the top are the 2 FMTs, and by the first week, their microbiome goes up to the diversity level of the donor, as you would expect, because that's what we just gave them. Whereas when you give them just 15 strains, that's not enough to fill the whole ecosystem, so it's more like a little patch. And you can see that the diversity there is quite a bit lower than what you see for the FMT, but by about 8 weeks, it catches up. So I think this is just different styles. If you're using a full spectrum community, you're going to give them the full diversity, whereas when you give a small community, you're trying to give them just enough microbes to be maximally efficacious and let the rest of the ecosystem develop. If we think about microbiome stewardship, just like we currently think about antibiotic stewardship, I think it's the same type of idea. You try not to give antibiotics to people that don't need them, because doing so reduces the diversity of the population on the planet and increases resistance. I think microbiome drugs ideally would be the same way. We give people just the microbes they need to make them better and let the rest fill in from the world around them. Because if we start giving babies at birth a full ecosystem, we end up making a small bottleneck of a few microbes that populate us all, and at some point we might regret that, just like we did with antibiotics. But, it's hard to say. This is the same type of plot here, but using a PCOA. The little triangles, let's see if I can use this thing. Yeah, these little triangles here are the donors. The blue is the fecal transplant, and the green is MTCO1. And as you would expect, the fecal transplant goes right up there with those triangles, but the MTCO1 kind of goes halfway and stops, because you're getting part of the donor's microbiome, but not all of it. So this is the main thing I want to try to convince you all to think about, which is to think about setting this thing up for your own lab or at your own facility. There's a little bit of details, but the main thing is that how long does it take to do all of this? There's the manufacturing, the regulatory, and the clinical trial. It all takes about 4 years. And 4 years is like perfect. That's like the generation time in science, right? That's how long your postdoc takes. That's how long your PhD takes. A little bit more than that, but more or less that's where it is, right? So in one generation, you could go from making a drug, seeing it through and analyzing the results. To me, that's more impactful than like, I sacrificed 300 mice and half of them got better. So what's the barriers to do this? The first one is always cost. How much is it going to cost you to do it? It costs about $100,000 for us to buy that equipment. That's enough money that I don't have it in my bank account, but on science terms, it's not that bad. Manufacturing was about $200,000. Most of that is for the salary of the person doing the manufacturing, but we do some amount of outsourcing of the safety assays. And then finally, the clinical trial cost is going to be, whatever clinical trial cost is. Very dependent on your country, how much is covered by your insurance versus how much you got to pay for. But for us, it was about $400,000. So not crazy high numbers, and I think, if anything, this is a little bit easier to fund, at least in the United States, than it is to do mouse work, because people would like to treat a human more than they would like to treat a mouse. So think about this for your own lives. The last thing is, it can be shorter than this if you don't have to start from scratch like we were. So we've provided all of our manufacturing protocols, all of our regulatory interactions, which are for the FDA, so I don't know what the thing is called here, but sure, it's a little bit different. They're all on this preprint if you wanted to download them. And we've built a lot of stuff over the course of the 10 years of running our lab. Sometimes people come visit, but this is the thing people are coming to visit the most. I don't know if people just all of a sudden want to take trips to New York, but so far we've had 4 people that want to come visit and see how to build one of these things. So if you want to come see us, come stop by. All right, so just a little bit about what this whole process looks like. The main way that these would be manufactured is we had this chamber that has 2 sides, and one microbe would be made on one side and another microbe would be made on the other side. And that was really the bottleneck, that once a week we would clean this thing, put 2 new microbes in there and go back and forth. And it takes about 2 months to get through the master cell banks, the drug substance, and the drug product. So this is not a huge amount of time, but in real life, people take vacations and things always take, equipment breaks, things always take longer than you want them to. So we want to make a lightweight version of this so more people are willing to try to do it, and also so that we can run lots of small trials rather than having these epic trials that determine our whole fate. I would like to have 2 or 3 trials going on at once if we could pull it off. And so that's where Monica Chaudhary comes in. She's a new postdoc in our group, and what she's been working on is trying to do parallel production. So she switched from having 2 different chambers to make 2 microbes at once to having 1 chamber to make as many microbes as she wants. That's the incubation chamber. And the manipulation chamber is where she harvests microbes and inoculates microbes. And the key to this has been moving to solid state. So a lot of you we have probably a bunch of microbiologists here, and you're going to go tomorrow and grow yourself a little bit of E. coli or something. You grab it, you stick it in your flask, and you stick it and shake it, shake it, shake it. The next morning it's ready to go. And then you usually put it on like an agar plate. And we don't think too much about the agar plate. That's just like selection and harvesting and stuff like that. But actually growing microbes on agar plates or on solid state media is an extremely efficient way to grow microbes, especially anaerobes. And it's very space efficient. So what we've ended up doing is moving almost all of the microbes to being produced on some type of solid media. And this allows us to save a bunch of space. So inside these chambers are these big, large trays that some old people at the Sanger here that maybe made those back libraries and stuff, probably made them on these types of plates. they're actually very good growing vehicles for manufacturing large amounts of microbes, and they don't take up a lot of space. And that allows us to move from this like 2 microbes a week, kind of slow prodding type of manufacturing, to making all the microbes sort of staggered over the course of 2 weeks. So it's about a fourfold increase in the time it takes us to go from a microbe to a drug. So we can currently do the working cell banks, the drug substance, and the drug products in 2 weeks each. So that gets us down to about 2 months, a lot more flexibility than to try to make different consortium. So why solid state? Well, one, it saves a lot of time. You can make a lot of microbes in parallel, but the two most important things for sort of real life are that the flexible harvest time is key. If you're trying to make 15 microbes, there's always a few microbes that don't behave very well. You know they should take 12 hours to get to peak performance, and then you want to harvest them, so you start at like 10:30 p.m. You're like, okay, so I need to wake up, be there at 6:30 in the morning. You wake up and they haven't grown. Or they wake up and they've grown like super thick and you have to start all over. The nice thing about the solid state is they tend to go a little slower, and then they stay at a happy point for quite a bit longer. So instead of having this like peak window where you can harvest, you have about a day. That makes it much easier to have a lot of things going on at once and to still have peak performance. The second thing that you don't really realize until you start doing this on a large scale, most of us grow stuff, we grow 5 mils or something, and then we spin it down. You can use any centrifuge you have around. Once you start having large amounts of water, it's hard to get rid of all that water, and it ends up becoming the slowest part of the whole process. So by going from these huge flasks with liters of media to scraping some microbes off of a plate. When you scrape the microbes off the plate, they're almost dry already, so you can just use a small microcentrifuge to do most of the processing, which saves a lot of time and money. If we look at the sort of how much the microbes like to grow in one way or the other, this dotted line, if you're on the top of the line, the microbe prefers to grow on solid state, and if you're below the line, it prefers to grow on the liquid. And you see almost all of the microbes prefer to grow on the solid state. And even all except for 3 of them, we're still making it on solid state, even when liquid is slightly better, just because it's so much easier to handle them. So of the 15, now we make 12 on solid state, and we make 12 on solid state and 3 by liquid. Okay, the other thing that you learn a lot about mice when you work with mice. You know, don't grab them too slow, be confident, otherwise they're gonna bite your thumb. With humans, you also learn about human behavior. I live in the United States. We have an interesting healthcare system where healthcare costs a lot of money. People like to make fun of us for that, but it's also very fast, which people can be jealous about. But the one thing that's weird about our healthcare, it costs a lot of money, but to the patient, it feels kind of almost free. You can buy a $10,000 drug or a $200 drug, and your copay is still like 10 bucks. So humans don't make decisions based off of money. They base the decisions more on convenience. So when we started our trial, Vowst had just become approved, but it wasn't available through very many medical insurers. So people would prefer our drug because it was what was available. But as time went on, or they could buy Vowst on the market for $20,000, so do you want free, or do you want, I guess it was probably $17,000. So free, $17,000 is a little hard decision. But once it became kind of feeling like free to everyone, nobody wanted our drug anymore, because you could have a colonoscopy or you could pay like a $15 copay. What do you want? $15 copay. I'd pick the same thing. So it became really clear that if we wanted to even go anymore in C. difficile infection, we had to move towards an oral formulation. But we didn't want to go with this whole like lyophilization, milling, the whole process that's going to take a lot of work to optimize. We wanted to find something that was liquid that would work just as well. So we tried different types of media, a whole bunch of different ones, but the one that's the simplest and ended up working pretty well is just modest amounts of glycerol are enough to keep the capsule from melting. Patients don't want to, you could tell them, swallow this, but don't wait more than 5 minutes or you're going to have a mush. People don't want that because people don't read instructions on the drugs. So we wanted to have a period of at least a couple of hours where the drug would be stable in their room temperature. And we found that a modest amount of glycerol is enough to keep a stable drug, and the microbes seemed to be happy enough in it. The other thing we needed was a way to put the microbes into these pills, so what we use is just a liquid handler to put microbes into each capsule, and we have this like fancy loader machine to turn them into capsules. So that leads us to our second trial, which we are hoping to start in the next few months. We've completed making working cell banks, drug substance, and this month we're putting together the drug product and putting it inside the pills. That will lead to our second IND with the FDA. And our goal with the next trial is to do a head-to-head with MTCO1 with and without a light bowel prep. It's unclear if doing a bowel prep to wash out antibiotics for C. difficile infection is actually beneficial or not. And if patients had to choose between do you want a bowel prep or no bowel prep, we're going to go no bowel prep. So we're trying to see how important is it to, and how much impact this has on engraftment. So we're going to try for 3 high doses, since we showed that the high dose seems to be a little bit better than the low dose, with or without a bowel prep. If you live in the United States and you know anybody with C. difficile infection, tell them to come our way, probably, hopefully sometime this summer. Okay, so this is all like doing basic science and translational discovery in an indication where we already know we can make some progress. So it's fast to learn, which is why we've been working in C. difficile infection. But we want to move towards other diseases so that we're go past the end of the beginning, but the beginning of it, the end of the beginning. So what other indications might we want to go after? And I think ulcerative colitis is the cleanest one, and it used to be the one I wanted to go after, but now I'm a little bit afraid of it because I'm not exactly sure what the next steps are. The top are some of the approved drugs. The bottom are the 7 largest FMT studies that have been RCTs. And if you look at the bottom, it looks pretty good, right? There's just a pretty good consensus, really only one major outlier where the trial design went in the opposite direction. That's the one by Senes Pell in 2024. But at the same time, that's one of the best designed trials, I would say, of all of these, and one of the best analyzed. And that's the problem with these fecal transplants is every person is getting a different drug, and it makes it hard to know why it works sometimes and when it doesn't work. So I really think we need to be moving much more towards defined consortium, because at least they're a failure. It's like, okay, those microbes didn't work, we're done with them. Whereas an FMT, there's a lot of reasons you can make excuses why it worked or didn't work. And this makes it hard to know what's the next steps. So I think we really need to be pushing towards a more hypothesis-driven endpoints that are single endpoints, or at least one dominant endpoint that you're aiming for. You can have some other friends if you want them. But what we typically have now is when people describe their microbiome drugs, they have all kinds of things they say it's going to do. Reduce biosis, Tregs, increase diversity, change the short-chain fatty acids. But which is the one that you think is the one that really matters for your drug? So I think we have to be more like a proper drug where you say, I'm going to try to change A to B. And if I get B and it doesn't work, then my drug didn't work. Easier said than done when we have multiple microbes and they probably have multiple mechanisms of action. But at the same time, if you're going to state a mechanism of action, I think we have to quantify it in a way like we do for C. difficile, where more engraftment seems to predict better response. So of the types of drugs that we could be thinking about for the future, these are the ones I think are the sort of categories. There's like restoring disease caused by depleted or missing microbes, which is what C. diff is. There's chemistry, like microbe A makes serotonin. Serotonin might be good, so I'm going to put that in there and measure serotonin changes. And then there's immunomodulation, which is my favorite, but also I think the hardest to know what the metric we're going to read out is. And then there's sort of more microbiome-inspired products, which is like taking parts of microbes and giving them to people. At this point in my life, I don't want to take on too much risk. I would like to build on victories. So our goal is to focus more on this restoring diseases in the cases where microbes are missing, to see if that, we know we can do that and we know we can measure it, does it make that person better? And the indication that we're interested in, although I think there are several of them, the one that we find the most interesting, because we're mostly interested in inflammatory bowel disease, is pouchitis. So I think pouchitis is an interesting target for an LBP. And I'm going to show you sort of our work in progress. This is going to be a little messier because we're in the process of trying to do all of this. But just show you how we think about developing our next drug and what we've got so far. And if you have ideas of things that we're messing up, please come talk to me. Or come on the run with me tomorrow morning and tell me then. Okay, so what is a pouch? So people with ulcerative colitis, when it gets to a terminal point, will have to have their colon removed. And when you get your colon removed, you can opt to have your small intestine sort of spun around on its end and turned into this little rectum that is called a pouch. And that allows them to have continuity and often makes them feel better. The problem is that in about 80% of people, that little pouch gets inflamed. And once, it's probably a tragic thing. You've been your whole life getting this serious disease, then all of a sudden you get the cure to the disease, and now you have a new thing called pouchitis. Most people respond to antibiotics, which I think is an important point. Like this is if we want to make a microbial therapeutic, it's really beneficial if the treatment to the disease is an antibiotic, because it shows that there's some microbial component to it. The problem is once people respond to antibiotics, they often relapse, and the more that they relapse, the more they end up on these chronic antibiotics, and it becomes a little bit like C. difficile infection where the treatment and the cause are both tied in with each other. So I think it fits the model of the type of drug that could work. Another interesting reason for going after pouchitis is that the pouch is different than the colonic microbiome. It's very sparse. So this is just looking at the density of microbiomes. There's the healthy, the ulcerative colitis, the Crohn's disease, and IPAA is just another name for the pouch. You can see it's by far the lowest in terms of its density, and if you just streak it out by CFU, you end up with about 1,000-fold less microbes per gram in a pouch than you do in a colon. From a manufacturing standpoint, that's quite attractive because it means we can make as many doses as we want for very cheap, because the biggest part of manufacturing is, can you make enough for a mouse? Sure. Can you make enough for like a however many stone human? Maybe not. So for these people, it ends up being a much less ambitious task to make the microbes. The other thing I like about pouchitis is that the features have replicated well across studies. There's a ton of studies on ulcerative colitis and Crohn's disease, and some things repeat across studies, but everybody's so different it's hard to say much. But in pouchitis, people only have like 5 to maybe 25 microbes, so the overlap across people becomes much higher. And if you look at the diversity as you go from right to left here, these are people that are not sick to people that are sick, they get progressive loss of diversity. And if we look at the microbes that are lost in people, the same groups show up and over and over again. This is just an example of showing that people with pouchitis have less Bacteroidetes than people that don't have pouchitis. This even replicates by culturing. If we take people with pouchitis and try to culture their microbes on an enrichment media for Bacteroidetes, oftentimes you find 0. Whereas in a healthy person, I don't think I've ever found a human with no Bacteroidetes in them that has an intact colon, but these people can actually have 0. So it becomes much clearer which types of microbes are missing. So what is our plan for designing a therapeutic for pouchitis? So our hypothesis is that these microbes are being lost by antibiotics, and what we want to do is to restore those microbiotics and see if it changes their chance of relapse. So from a clinical standpoint, what we want to see is when we restore those, does the duration to their time of relapse go down? Can we prolong how long it takes someone to relapse by putting back their microbes? But the question from the basic science standpoint, which is where we are right now, is how do we pick those? There's all these debates, and it probably happened at this conference. People will debating what is the healthy microbiome. It goes on for hours and hours, and then it probably gets even more extreme. But I think for the pouch, it's not so much debatable because the universe of microbes that they have in their pouch is much less than what are in healthy people, and it makes it much easier to define what is the healthy pouch. So this, as I said, was work with Maya Kaya, who's the MD at Mount Sinai, who's just done an incredible job of building all these human cohorts that we're using to study the pouch. So I don't want to get the impression that this is easy. Now we could say C. diff is easy, even though at the time it's never easy. It's only easy after it's solved. But if you think about where we were for C. diff, pretty much every FMT donor works for C. difficile infection. If you look at what's happened in the pouch so far, I would say it's mostly a graveyard. There's a meta-analysis of about 130 people or so that have been treated with pouchitis. What I'm showing here on the left is the largest RCT, which is not that big. But if you look at this RCT, the placebo's on the top, so the bad guys won. So people got treated with stool, and they ended up not statistically worse, but it looks worse than the people that didn't get treated, which is a tricky sign for if you're going to go after this as your first indication or second indication. But recently there was a paper by Kusgaard et al. Where they treated 3 people, and they treated them with pouch stool, healthy pouch stool. And I think this is critical. You want to match the ecosystem with the ecosystem. If you put healthy donor stool blended up in the pouch, you're probably overdosing that person by at least 1,000-fold with a bunch of microbes that their pouch is not used to seeing. But if you treat someone with pouch stool, you have a better chance, I think, of some success. And what Kusgaard found, unfortunately, this study only had 3 people, but all 3 people went into remission and had some amount of response. So this leads us to where we are right now. We have 3 cohorts that we're trying to use to study how do we pick the best set of microbes for developing a pouch-based drug. The first cohort are people that we're looking at how they progress through their pouchitis. That is, they start healthy, they get antibiotics, and they end up going from these different stages of like acute pouchitis to chronic pouchitis, and we see which microbes are lost. The second cohort we have are some people that have these healthy pouches and they don't need any drugs. And we're trying to see over time which microbes are those people able to keep, so we can know what are the durable microbes that we might give to someone to give them a therapy that would replace their missing pouch microbiome and stay around for a while. And then the last thing that we have going on is a trial similar to this Couscard et al., where we are giving people with pouchitis healthy pouch stools to see which microbes can transmit well and which ones engraft in the people that get better. So for the sake of time, I'm only going to show the middle one, but we have all three of these going on in parallel. So what is the stability of a healthy pouch? And I would guess that no one here knows, because I don't know, and I don't think there's that many people even on the planet looking at the pouch. So what have we done? We've taken 7 people whose pouch was stable over 1 year, and they had no antibiotics. Most of them have been stable for several years. We did high throughput culturing at the 0 time point and at the 1 year time point, and we did metagenomic strain tracking and just genome-based strain tracking. And that's on the bottom here. Each row, if the color goes across the row, the microbe was found at 2 time points, and if it's colored at one side and not at the other, the microbe was found to be different at the 2 time points. On the bottom is the numeric summary of this, which is just the Jacquard index. 0.5 would mean half of the microbes were stable. And if you look at those first 4 people there, not so stable. It's like over 1 year, even though those people didn't get sick, almost a completely different microbiome. Both of the microbiomes look healthy in the sense that they have a lot of the microbes you would expect in the healthier pouch, but totally different microbes over time. Whereas the people on the right side, those 3, had much more stability over time. If we compare this to the trajectory of a healthy colon, this is some work from ages ago, back when I was a postdoc at WashU in Jeff Gordon's group, and we had about 30 people when we would look over the course of a week, 2 weeks, a year, etc., and see what proportion of the microbiome was stable. We found that about 60% of healthy people's microbiomes were stable, but even at 1 year it was probably more along the lines of 70. And if I just overlay these pouch data on top of this, you can see it's not as nice as a healthy colon. That there's even the best people are quite a bit different than the healthy colon, and the 4 worst people are much lower. And these are for people that have been healthy the whole time. So I think this is going to be something to set expectations for how often we need to dose people to keep their microbiome the same. And I think the answer is it's not going to be once in a lifetime like it is with C. difficile infection with these people. Their transit time is quite fast, so instead of making 1 or 2 bowel movements a day, they make more like between 6 and 12. And there's no like appendix for the microbes to hide in, so you can imagine it's harder for microbes to take a nap. They pretty much have to be growing their whole life if they want to not get washed out of this ecosystem, which might explain why microbes are lost so much from the pouch. The other thing that we notice is that there are some microbes that are pouch dominant and microbes that are colon dominant, and some microbes that are pretty common in both. So on the x-axis here are the pouch cultures from these 7 different people, and then we cultured from 7 different fecal transplant donors just to see how many times we culture each species. Each dot here is a different species. And in the boxes are species that were cultured a lot of times from the pouch, but not from the colon, and a lot of times from the colon and not from the pouch. So I think just the idea that you would take colon microbes and put them in the pouch is probably not realistic. I think these microbes are evolving to stay in the pouch because they have a faster ability to grow. Okay, almost done here. So this is our plan for pouchitis. This is more for me than for you because I can look back in 2 years and see how close we got to maintaining this timeline. Always drifts a little bit more than you would hope, right? Everything always takes too long. But our hope is in 1 more 4-year period to go from these observational FMT type studies to defined therapeutic in testing it, hopefully starting sometime next year. Okay, so back to our first question. Is microbiome research at the beginning of the end or the end of the beginning? And I think an important thing, especially for the junior people here, is that we decide that. We decide that by the decisions we make every day. You can do science for the pure beauty of doing science, which is great, but then you can't expect your friends and family to want to fund the science if it's like looking at the microbiome of tadpoles in cold swamps in Scotland. It's very interesting. It makes for good, interesting conversations. But people in general want to make themselves better because we're like selfish human beings that want to feel not sick. So mix your cool basic science work, which I like to do too. I showed you about half of what we do is this type of stuff, and the other half is just like trying to figure out what the future looks like. But I would encourage you to don't just do basic science. Think about how you can have an impact today. You can always be like, oh, in the future, I'm going to translate this stuff. No, translate it right now, because if you don't start right now, everything always takes about 10 years, and you're going to be grown up and know stories to tell people about the drug that you developed. So if no one's shooting on goal, no one will ever score. So think about taking some shots on goal. And I think the whole setup is not that expensive. And I think the rules around mice are pretty complicated in the UK. From what I hear, like, ooh, boy, sounds like it's pretty hard working on mice here. It might be easier to do a human study than it is to do a mouse. We built a germ-free mouse facility. I would say that running our germ-free mouse facility is more work than our human manufacturing facility. So think about that. It's easier to recruit the mice, but other than that, I think the humans is more fun, and so think about doing that. So with that, I'm happy to take your questions. The people in pink here are the people that contributed the most, but it's a group effort with lots of clinicians and lots of scientists, so thank you for your time.
Thank you very much for this amazing talk. So we are now open for questions. If anyone has a question, please raise your hand and the microphones go to you. In the meantime, I have one. So I was wondering, especially for the pouch patients, how do you plan to control for bacterial translocation when you are picking the species you're going to do in your consortium?
What do you mean by translocation?
Because you have a pouch, right? You're going to have lesion in the gut barrier. Some bacteria can attach to the epithelium and then can translocate to the patient and make more inflammation and provoke more inflammation and then solve it. Can you control for that? Like choose those could adhere more or less? Or
yeah.
Like
I mean, I think one of our design criterion for the C. diff was that we didn't use any facultative aerobes, because then you can't get like a BSI or something like that. For the pouch, it's a little tricky because they do have more aerobes in them. In terms of translocation, I think part of that part of the reason I think the active group did worse than the placebo in the fecal transplant trial is that, like, you overdose someone and you can have like inflammatory response to this. It seems like we need some microbes for tolerance, but too many is like inflammatory, right? So I think getting the dose right is critical. And I also think if you have people that have been healthy for multiple years and you look at their stable microbes, those are less likely to be inflammatory because they've been in the person for a year and the person didn't have any sickness, right? So a lot of it's empirical.
Can you correlate IgA against this bacteria?
It's a good idea. We haven't done that. This is smart. We should do that.
So we have a question over there.
Thank you. That was inspirational. And I work in a completely different microbiome space, the women's health. And I suppose my question to you was, from the significant amounts of data you've got on donors that confer engraftment success and strains that confer engraftment success, what functional data, what predictors of engraftment success has the field had in terms of looking at strains that do durably colonize? And are those strains, do they just reset the ecosystem, or do they hang around and stay there long term? Those were the sort of the obvious questions, and whether durable colonization requires antibiotic pre-treatment. I know you spoke a little bit about whether you needed. So a lot of questions that we could take into another field. Thanks.
Yeah, so antibiotic pre-treatment, we don't know if the only data out there without antibiotic pre-treatment, I think, shows that antibiotic pre-treatment is beneficial. Here we're treating people with C. diff, so they already are getting antibiotics as standard of care. Picking which microbes engraft well or not well. So one of the studies that we've collaborated with is the FOCUS trial in Australia, and they pooled like 4 to 7 donors together. And it was really interesting in there that in pools they would mix which donors were with each other, and certain strains from certain donors would always get in. Even if there was like 5 B.O. viruses, the same one would always get in. What there was genetically about that one that made it sort of more infective, I'm not sure, but it seems there are properties of strains that drive whether they can get in. But so far our work is really empirical. And our design of like which microbes need to get in for which disease to do well, also is quite empirical, which I think is really all we have at the moment. Because we do functional work in mice. The problem with the mouse experiments now is that we tend to use kind of like microbiome virgin mice. Like they haven't had that much infections in their life. And I think a lot of the phenomenon that we see of induction of different immune cells might be different if they like had norovirus, COVID, and then your microbe, right? But instead we give them like, this is the first stimulation you see, and it's like this very pure immune response. So I think bringing some realism to our mice models in terms of an infectious history like we all have is going to be critical if we're going to pick these kind of strains properly using mice.
Great, thank you. Jeremiah, that was superb. Thank you. Kicked off the conference in a brilliant way. Thank you. I'm most interested, I guess, in the pills that you talked about. So obviously delivering these anaerobes is problematic because they will keel over when we expose them to oxygen. So I just wondered if you could maybe expand a little on, you know, the viability times when they're inside this pill form, and whether or not that varies from strain to strain based on things like oxygen sensitivity.
Yeah, so for sure the pharmacutes are less happy than everything else, as is always the case. I mean, part of the next study is that we have colonic delivery, which is going to be the best there. We essentially wake them up, and within 1 hour, they're sitting in the colon probably happy. And part of doing the same 15 microbes orally is to see that. We also have some collaborations going on with some of the fecal transplant groups where they've given the same donors by enema, by upper endoscopy, by lower endoscopy, and by pills to see what's kind of being lost. That is a worry. We're trying to overcome that by dosing multiple times and hoping for the best. We have done stability. You know, these microbes didn't get transferred rectal. They got transmitted rectal-oral, right? So we know they can do it. And when you get a dose from your sibling or something when you're a little kid, they're not giving you a 10 to the 9th, they're giving you 10 to the 2. So somehow it can happen sometimes. I just think it depends on how much space there is in the ecosystem for it to fill in and what's the MIC. We've tried to do some type of experiments where we label lots of microbes and we see what's the bottleneck of how many is the minimum dose for each microbe in mice. But it'd be nice to do that in humans. Just the problem is too expensive. But it is a worry.
We have a question over there.
Oh, thank you. Thank you for a very interesting talk. I was interested to know, do you also see any functional difference in the microbiome in the healthy pouch versus non-healthy, on top of the taxonomic difference that you see?
So I would say that the pouchitis is a subset, but a very, very small subset of the healthy pouch. A healthy pouch tends to have close to 20 bacteria, and the pouchitis pouch tends to have like 2 to 6, and most of them will be facultative aerobes. They're getting hammered with antibiotics a lot, right? And they're never off antibiotics long enough to know would it have recovered on its own at some point. So even when they're off antibiotics, they tend to have these very sparse microbes. There are the same species of bacteria in the healthy pouch and in the sick pouch, and I don't know if, like R. novis, for example, is in almost all of the people with pouch. In regular people, maybe it's in 10%, 20%, and pouchitis, or in pouch people, it's like 100%. But do the ones that have pouchitis have a bad one, and the ones that don't have a good one? That I'm not sure. And it dictates kind of what we give to the people, because do we want to try to put the good one to the bad? And this is where our knowledge of the microbiome is really fuzzy, and it's hard to know what's the right decision, right? Do you avoid the bad ones, or was like this less bad, so you want to try to use it to push out? Also why we sort of avoid E. coli when we give people drugs, but you could imagine scenarios where like giving people a lot of good E. coli might push out the sort of antibiotic resistant ones. But it's a good question, I just don't know the answer.
Some question there.
Thank you, great talk. I'm Sasha Shatnakova from Groningen. I would like to have some more insights maybe about how you selected these bacteria. And in particular, I'm interested from 3 different sub-questions. One is, when thinking about beneficial bacteria, I would expect to see Faecalibacterium prausnitzii there, but I think it was not. And second, people might take these along with some probiotics that have known and established bacteria. How does it have an effect? And the third one, there are a lot of genetic variations in bacteria. So from strains, you can select those that have certain number of genes or do not have certain cluster of genes. I'm curious, how did you come up with these 15 actually? And,
the 15 is just empirical, because we know for C. diff that every donor works, so it shouldn't matter too much which donor you pick. Why not F. prausnitzii? because not everyone has that bug, and it's a pain in the butt to grow. I do think in IBD, it's a smart one to have. People with pouchitis, or excuse me, people with healthy pouches sometimes have it, but not often. I don't understand how it can grow in the healthy pouch, but it seems to be able to. But that doesn't seem like a very anoxic environment. In terms of the SNPs, part of the reason, so I don't know, I think our field is a little bit sloppy in that the diversity is so high, you could always pinpoint some type of gene to explain a phenomenon that you have, because we have too many variables. And part of the reason for trying to encourage people to build a system like what we have is that you can test these hypotheses. Like, I don't know, it could be that you need this SNP, you might need this gene. Why don't you try it and see if it matters rather than just telling a story about it? So for us, we've just been really empirical, using fecal transplants as the driver, as other people have been doing. But ultimately, yeah, that's the level of mechanism we would like at some point, which is like, you need metabolite X from microbe X, and then you get better. But that's not where we are right now.
I have an online question.
Yeah, so this is from Monica Wong. What's the role of lifestyle, for example, diet and exercise, in the recipient? Does it affect the persistence of transplanted strains?
Yeah, for C. difficile, it doesn't seem to, because everybody has stability. I would guess in the pouch it would have much more of an impact, because you're really trying to maintain a few microbes and they are surviving on like very rapid growth. We are interested in the potential to use symbiotics, but my experience recruiting humans into diet trials has been a real pain in the neck. So people say they want a diet, but then when you offer them a pill, they prefer the pill. So we're going mostly with pills.
I've got a question which is related to sort of the live biotherapeutics that you talked about. So we're involved in trials looking at fecal transplantation of patients with advanced chronic liver disease. So these are patients with cirrhosis, pretty sick, needing a liver transplant, and we're fecal transplanting them at the moment in the UK. That's a 300-patient trial, big study using essentially encapsulated stool. We're already looking, obviously, at the next generation of live biotherapeutics. My question to you is around this, the journey to get from FMT, which for us is substandard, suboptimal, and has all sorts of problems associated with it, to getting to sort of more defined consortium. And how do we get there at a more rapid rate, and what are your thoughts on that?
Yeah, I would say you're in the perfect position to do this. Of all the things that if I was going to put a ranking, I would put liver transplant somewhere in the top 10. Like, those people have depleted, crazy-looking microbiomes. And you can empirically look from these people in your trial. The trick, the trial's probably a long ways in. The key thing is having not too many donors. So if you can have just a few donors going to a lot of recipients, then you can look at the recipient outcomes and what engrafts and learn a ton from just that, right? So I would say do the same type of thing we're doing. Grow out the microbes, and then you can just try different, either one large community or a bunch of different small ones in a trial that would be much easier to run. And at least in the United States, like the regulatory process and all that has been far simpler with defined consortium than with like, we're gonna mix up stool and give it to people. So I think you're already past the start line, and you're really, really close to being able to make a therapeutic from what you got.
Sorry, I'm here. I have a question. Thank you very much for this talk. I'm very curious about your opinion on the recipient effect of FMT engraftment, because you were talking about like bacteria that engraft more. So what is your opinion on, for example, those concept of gatekeepers, for example, that a person can have a microbiome that is not enabling an FMT?
I think that's a good question. So far, we're going after people with the most destroyed microbiomes, and I think it's less of an issue there because they've people that have been on chronic antibiotics. If you just give this straight to like a healthy microbiome, we're going to start to do some work like that, and I think there will be much more endogenous microbes that are like, no, we're already there. And I think this is one of the challenges with ulcerative colitis type trials, is that those microbiomes are not completely depleted. Actually, it's a pretty decent looking microbiome in people with IBD. It's just slightly different. But which ones block which ones, I don't know. And that's the type of thing that probably works out better in a mouse, where you can do, one versus many and many versus one type of experiments, than in humans, where every recipient has a different microbiome, so it's hard to quantify which ones are inhibiting your microbe.
Is there any more questions? Question from the virtual audience? So, thank you very much for the great talk.